Hi, my name's Margie Dangerfield. I'm a registered nurse, midwife, and independent Zinzino partner, and I've helped almost 3,000 people using Zinzino's test-based nutrition. In this video, I'm bringing you an industry leader and an expert in their field who are integrating Zinzino's personalised test-based nutritional approach into their clinical practice. So, if you're ready to move beyond guesswork and elevate your health and your patient outcomes with precision-driven strategies, you're in the right place. Well, welcome everyone, and thank you so much for joining us tonight. I am absolutely thrilled to have Dr. Martina Torrissen with us. Thank you so much for joining us, Martina.
Thank you very much for having me.
It's been a high of activity since the launch of the gut test in Oslo, and there's been a lot of discussion around the test. And I know that a lot of us listening may have even taken the test, so we're absolutely excited and privileged to have you here because, you know, this breakthrough in gut health test is a world first. So tonight, we're going to have a presentation from Martina, and then we'll open it up for question and answers. So if you do have a question, please feel free to type that into the chat. And at the end of the recording, end of the presentation, we'll stop the recording and we'll have maybe 10 or 15 minutes for Q&A. So welcome, everyone. I'd like to introduce Dr. Martina Torrissen. She's a nutrition and bioscience researcher whose work explores how diet and microbial metabolism shape immune, metabolic, and gut health. With a background in molecular nutrition and lipid metabolism, her early research provides new insights into the biological functions of very long-chain omega-3 fatty acids and how these unique lipids contribute to cellular structure and tissue integrity. Building on this mechanistic foundation, her current research focuses on the gut microbiome axis, particularly the role of tryptophan metabolism and microbial metabolites such as IPA in maintaining gut barrier integrity and systemic balance. She has led the scientific development of the gut health test, the first of its kind blood-based analysis designed to quantify microbial activity and reveal how diet and lifestyle influences gut and metabolic health. Martina holds a PhD from Norwegian University of Life Sciences, and her work has been published in leading scientific journals, including nominations for Paper of the Year, awarded in the British Journal of Nutrition. So thank you, Martina, for being here, and I'm very much looking forward to the presentation tonight.
Thank you. Shall we just get started then? Today, we often talk about it in different parts. We talk about the gut and the brain and the immune system, and we often talk about it as if they are separate departments of the same body. But the more we learn, the clearer it becomes that the human body doesn't work in parts. It is a system and it is a network of constant communication. And the gut sits right at the center of this network. So even when most people think of the gut, they think of digestion. So food goes in, nutrients get absorbed, and then waste comes out. And while this is true, it is just the surface layer of what's really happening, because the gut is our largest immune organ. It houses around 70 to 80% of our immune cells. And it is also home to trillions of bacteria, which constitutes our gut microbiome. And this forms an entire ecosystem living inside of us. And these microbes, they don't just digest our food. They also talk to the body. And they do this through chemistry, through the molecules that they produce as they break down what we eat. And these molecules, they are called metabolites. And they move beyond the gut wall and they send messages that affects our metabolism, our immune system, and even our brain. So the gut is not just a digestive tube, it is a communication hub. And you can think of it as a central interface where the outside world, meaning everything that we eat, breathe, and feel, is translated into signals that also guides our inside world, meaning our physiology, our mood, our energy, and our resilience. So over the past 15 to 20 years, new DNA technologies have given us the gift where we are able to map out which microbes live inside of us. And this led to an explosion of microbiome research and thereby also this new way of thinking about health. So for a while, the main question was all about who is there, which bacteria are found in people that are lean versus people with obesity, and which ones are more common in those with gut disorders, or even in those who stay healthy as they age. And these are all very exciting questions, and they reveal very interesting insights, but they've also eventually revealed something that's also interesting, which is that two people can have sometimes nearly the same composition, yet different health outcomes. So that led to new questions, which is not just about who is there, but also what are they doing? And this is where metabolomics has changed the game. So instead of looking at microbes as names on a list, metabolomics allows us to look at what they are producing. So the chemical fingerprints of their activity. And it lets us see the function of the microbiome and how those trillions of bacteria are influencing our biology. So in a sense, you can think of these metabolites as the language of the gut. They are how our microbes are communicating with us. And when this language shifts, so when certain messages go silent and other ones become louder, it can also change how our whole system behaves. So when we look at this communication system from a distance, we can see how profoundly interconnected it is. The gut, it talks to the immune system every second, and then the immune system feeds back signals that also influences our microbial activity. And our brain listens to all of this through the vagus nerve and also through the molecules circulating in the blood. So it is evident that the gut is not a standalone organ. It is a systems organ, and it is a biological crossroad where immune, metabolic, and neural networks are constantly communicating. And when this network is balanced, we see stability across multiple systems. We see steady energy, balanced immune tone, and better mood regulation. But when it is disrupted, this imbalance echoes throughout the body, in the liver, the brain, and even in how we age. And this is where tryptophan becomes such an elegant example of systems biology in action. Tryptophan is an essential amino acid, which means that we have to get it from our food. But what happens to it afterwards depends on the conversations between our cells and our microbes. And you can think of tryptophan as standing at a crossroads with three possible roads ahead. And the first one is the kynurenine pathway, which is the main route, and it handles around 90 to 95% of tryptophan. And this pathway is essential. It produces NAD+, which is a molecule that every cell needs for energy. But it is also the body's stress response. So when immune stress or signaling rises, enzymes like IDO1 or TDO will open the gates wider, pulling more tryptophan into kynurenine. And if this stays active for too long, it can create a biochemical environment that fuels oxidative stress and biological strain. Now, the second road is the serotonin pathway, which is a much smaller road. It handles just about 1 to 2% of tryptophan traffic. And here tryptophan becomes serotonin, which affects mood, sleep, and gut movement. And then we have the third road, which is the one that interests us the most, and this is the microbial indole pathway. And this is where our gut bacteria take tryptophan and they turn it into compounds like indole propionic acid, or IPA. And this is not just a byproduct, it's a signal of cooperation. So when this microbial pathway is active, it tells us that the gut environment is healthy. The right microbes are being fed, immune activity is well balanced, and the body and microbes are in harmony. So in other words, when the system is under stress, tryptophan will flow more heavily into kynurenine, but when the system is balanced and nourished, it flows into indoles like IPA. So I like to use this traffic light metaphor where we can think of tryptophan as arriving at a busy traffic intersection. And then a healthy diet and microbiome diversity turns the traffic light green for the protective indole road, versus heightened stress or immune signaling turns it green for the kynurenine highway. So let's look a bit more on IPA, because IPA is one of the most fascinating compounds discovered in recent microbiome science. And it is made exclusively by certain gut bacteria when they have the right conditions, and particularly when they're fed dietary fiber. And the effects of IPA reach far beyond the gut. But first of all, IPA has been shown to strengthen the intestinal barrier, which is this thin, selectively permeable layer that keeps unwanted molecules out and nutrients in. But it also supports balanced immune communication, helping immune cells maintain normal activity. It acts as a powerful antioxidant, protecting cells from oxidative stress. And it has even been shown to influence our metabolism by helping to regulate blood sugar. And what's interesting about IPA is that it doesn't just reflect a healthy gut, it helps to maintain it. It creates a feedback loop where the more IPA your microbes produce, the stronger the barrier becomes and the more stable the system stays. So from this, you can think of IPA as a guardian molecule, and it's a molecular signal of cooperation between us and our microbes. And diet is one of the strongest determinants for whether tryptophan flows into the kynurenine or the indole road, and fiber is the single most important driver. We know that our ancestors, they used to eat well over 100 grams of fiber per day. But today, most people in industrialized countries, they get less than 20. And this shift has completely reshaped our microbiome and with it, our metabolite balance. So when we eat a low fiber, high fat, typical Western style diet, our microbes, they lose their preferred fuel source and they start breaking down mucus and protein residues instead, which produces metabolites that are associated with heightened immune activity. And at the same time, immune signaling then becomes more active, which again pushes more tryptophan towards the kynurenine pathway. But when we eat enough fiber, the opposite happens. Our microbes will ferment fiber into short-chain fatty acids like butyrate, which helps to maintain balanced immune signaling and to regulate IDO1 activity. And this frees up tryptophan so that more can flow into the microbial indole route and produce more IPA, and thereby fewer of these stress-associated metabolites. And of course, fiber doesn't act in isolation. It also interacts with our fat quality, so the omega-6 to 3 balance matters, and it also interacts with polyphenols, which acts like microbial selectors that enriches the species that produces more of these beneficial metabolites. And it also interacts with stress, sleep, and physical activity, all of which influence our immune tone and the enzymes that will govern tryptophan metabolism. So health in this light, it's not about single nutrients, but it's about coherence and how our diet and lifestyle are working together to shape this network of signals. So to understand what really shapes IPA production, I want us to look a little closer on its biggest driver, which is fiber. And this figure here shows how fiber is a game changer for tryptophan metabolism through the mechanisms of how it influences it. So first, fiber slows down absorption. So more tryptophan can then reach the colon, where oxygen levels are lower and indole producing microbes can thrive. And then second, when fiber is fermented, it produces the short chain fatty acid butyrate, which also helps to maintain this anaerobic low oxygen environment and also modulates IDO1 expression and immune tone, which means that fewer tryptophan molecules are directed into the kidney pathway. And third, fiber feeds beneficial microbes, which supports these cross-feeding interactions that promotes the growth of species that are capable of producing indole metabolites like IPA. So with fiber, we get less tryptophan lost to the stress-related road and more of it directed into the protective indole road. And this is also why fiber is such a powerful driver of resilience. And we see that in population cohorts and intervention studies, they all indicate the same pattern. With higher fiber intake, this aligns with higher circulating levels of IPA in the blood and more favorable tryptophan ratios. And this is not just in theory, so I want us to now look a little closer on what this balance means in real-world conditions, starting with the liver. Because in the liver, this balance makes a very clear difference. And you can see in a healthy state, certain gut bacteria will convert tryptophan into indole compounds like IPA. And IPA activates a receptor called the aryl hydrocarbon receptor. And this is a molecular sensor in the gut lining that detects signals from our microbes and helps to keep the immune system balanced. And when the aryl hydrocarbon receptor is activated by IPA, it has been shown to strengthen the gut barrier by promoting the renewal of our intestinal cells and the production of tight junction proteins that helps to seal the gut wall and therefore makes the gut barrier stronger. And it also helps to maintain balanced immune signaling and normal immune tone. So the result of this is that we get fewer unwanted molecules leaking into the bloodstream, we get more stable liver signaling, and improved fat and energy metabolism. But in metabolic liver disease, when the gut microbiome loses its diversity and indole production drops, the opposite happens. The barrier weakens, the bacterial toxins leaks through, and immune signaling in the liver becomes overactive, which leads to fat accumulation and tissue damage. And in the gut itself, this contrast is just as striking. When the microbiota is balanced, like I said, tryptophan is then converted into indole compounds like IPA, which can activate this aryl hydrocarbon receptor in the gut lining. And this activation of this receptor by IPA strengthens the barrier by tightening the cell junctions and supports immune balance that keeps inflammation under control. But in inflammatory bowel disease, when dysbiosis sets in, the microbes that produces these beneficial indoles are reduced. And as a result, more tryptophan is then diverted into the kidney pathway, which is driven by heightened immune signaling. So the gut barrier then becomes more permeable and immune signaling becomes overactive. And this creates a feedback loop that sustains biological strain and also disease activity. And these same principles apply as we age. Over time, the gut microbiome often loses diversity. So this means we have fewer of these beneficial bacteria and a drop in protective metabolites like IPA. And with less IPA, the gut barrier weakens, immune signaling will shift, and low-grade immune activation becomes more common. And this is a process that is often referred to as inflammaging. But studies of healthy centenarians shows that not all aging follows the same path, because in these we see something very interesting, which is that they often maintain a diverse, balanced microbiome. They continue to produce indole metabolites, is consistently associated with higher circulating IPA levels, and they preserve stronger barriers and immune function. So, in other words, aging is not necessarily an inevitable decline. It is deeply influenced by the state of our microbiome and the molecules that it produces. And these same imbalances that affect our gut and our metabolism also reach the brain through what's known as the gut-brain axis. So when the system is in balance, microbial indoles like IPA helps to protect the blood-brain barrier, supporting healthy immune signaling and maintaining neurotransmitter balance. But when the gut is imbalanced, the barrier becomes more permeable, immune signaling becomes heightened, and the kindred pathway metabolites will build up, which are processes that can affect our mood and cognitive performance. And this is also why gut health is increasingly being linked to brain and metabolic resilience. So you've seen across all these systems in the liver, the gut, the brain, and even aging, the same pattern emerges. When tryptophan is channeled into microbial indoles like IPA, we see protection. We see stronger barriers, balanced immune responses, and greater overall resilience. But when more tryptophan is diverted into the kindred pathway, we see the opposites. We see weakened barriers, heightened immune activity, and lower adaptability. So what I want you to take away from this is that this balance is not fixed. It's something that we can influence. And diet, fiber intake, sleep, and exercise, as well as microbial diversity, all shape how these pathways flows. So in other words, the way that we live and eat, it doesn't just feed us, it also steers the chemistry that connects our gut, our metabolism, and even our minds. And by resilience here, we mean tighter barriers, calmer baseline immune activation, and steadier metabolic control, which are measurable hallmarks of a system that adapts and recovers efficiently. So now that we know that this balance between indoles and kindred isn't fixed, it's something that we can shape through what we eat, how we move, how we sleep, and how we manage stress. The next question is, how do we measure this balance in a way that is practical and meaningful? How can we actually see this? How can we see whether our gut and metabolism are moving towards resilience or drifting towards strain? And this is also where measurement becomes powerful, because for decades now, we've been able to see the microbiome, but not as easily its function. We've been able to catalog the bacteria, but not as easily measuring what they produce or how those molecules affect us. But now with the gut health test, this is much easier and more accessible. So by measuring just 3 key metabolites in the blood, which is tryptophan, kindred, and indole propionic acid, we can get a functional snapshot of how this system is performing. So tryptophan shows the available substrate, which is the fuel at the crossroads. You can think of it like that. And kindred reflects immune pathway activity and overall system load. And IPA reflects microbial health and cooperation. And from these, we can derive ratios that act as systems indicators. We have the kindred to tryptophan ratio, which is an indicator of immune engagement, or our immune stress indicator. We have the IPA to tryptophan ratio, which is the gut microbiome efficiency marker, showing how efficiently the microbiome is turning dietary precursors into this beneficial signal. And then we have the IPA to kynurenine ratio that shows the overall balance between protection and stress and is therefore our microbiome immune support marker. And when we combine them, we get the gut health index, which serves as an integrated snapshot of whether your system leans towards resilient or strain right now. So this is what we measure with the gut health test. And here you can see an example from the test report and how it will appear to the user. And I just want to emphasize that these zones, they are directional indicators. They don't diagnose disease, but they show you where your biology sits along the continuum from strain to resilience. And this approach keeps interpretation firmly anchored in preventive health, reflecting biological shifts observed in healthy populations and not clinical thresholds. So, in other words, the results, they help you guide understanding and action and not necessarily classification of disease. So, now that you've seen what the gut health test measures, I want to take a few moments just to walk through how to read the results and also how to interpret them. Because the goal isn't just to look at the numbers, but also to understand what they mean, what they say about how your microbiome and your metabolism are interacting. So when you open the report, the first thing that you will see is your gut health index. And it combines all the key ratios together with your total IPA level to show the balance between microbial support and immune metabolic activity. And you can think of it as an overall summary of a complex system. So if your score is in the green, high or medium high, that means that your microbiome is showing strong protective activity and relatively low immune tone. But if it's in the lower end, it doesn't necessarily mean that something is wrong, but it simply means that your biology is showing room for improvement. Because you should remember that this test doesn't diagnose disease, but it describes where your system currently sits along the continuum from strain to resilience. So the first specific marker in the report is IPA, and it's exclusively microbial, which means that your human cells cannot produce it. So its presence reflects an active microbial metabolism and a well-supported gut environment. And a high IPA value, like you see here, means that your microbes are actively producing this beneficial metabolite, which is a great sign of a nourished and fiber-supported gut. A moderate IPA value means that there is some microbial activity, but there is still room to strengthen it, typically by increasing fermentable fibers and polyphenol-rich foods. And then if you have a low IPA, this can indicate lower microbial support, which is often likely from low fiber intake. The second marker is the IPA to kynurenine ratio, or the microbiome immune support. And this ratio reflects the relative balance between the two pathways of tryptophan metabolism, which is one driven by the microbiota producing IPA, and one driven by the host's immune and stress-related enzymes producing kynurenine. And a higher ratio here indicates that microbial activity is proportionally stronger relative to immune-related metabolism. And this trend, seen in scientific literature and also confirmed in our internal data, it tends to align with higher fiber intake, favorable omega-3 balance, and a more stable immune profile. But a lower ratio signals that immune-related tryptophan metabolism is relatively more active. And this is a pattern that we often observe under conditions of immune activation, psychological stress, or reduced microbial support. So I just want to note that this ratio, it doesn't imply that one pathway suppresses the other, but it reflects the direction of the metabolic emphasis at the given time. And higher values suggest the system is leaning towards microbial cooperation and resilience, while lower values indicates greater immune engagement. Then we have the IPA to tryptophan ratio, or the gut microbiome efficiency marker. And this ratio shows how efficiently your gut microbiota converts tryptophan into IPA. A higher ratio here indicates stronger microbial conversion activity relative to tryptophan availability. And this is a pattern that is typically seen in individuals with higher fiber intake and greater microbial diversity, which are factors that are known to promote beneficial indole production. And a moderate or low ratio suggests that less tryptophan is being channeled through this microbial route. And this can occur with low fiber intake, reduced microbial diversity, or periods of physiological or immune stress that will redirect your metabolism. So this ratio reflects how active the microbial branch of tryptophan metabolism is under current conditions. It doesn't indicate an absolute rate or efficiency, but it's rather a relative signal of your microbial activity that is both in scientific studies and also in our internal data, tending to trend upwards with healthy dietary and microbial patterns. And then we have the kynurenine to tryptophan ratio, or the immune stress indicator. And this ratio describes how much of your available tryptophan is being converted into kynurenine, which is the route that becomes more active when the immune response enzymes are engaged. And a higher ratio here reflects stronger activation of this pathway relative to tryptophan availability. And this is also a well-established marker in research, and it is known to rise during periods of immune activation or oxidative stress. So a lower ratio indicates calmer immune tone and greater availability of tryptophan for alternative metabolic routes, including the microbial indole production. But if overall tryptophan levels are low, the interpretation changes and it signals reduced substrate availability rather than pathway activation. So this ratio is not diagnostic of disease, but it provides a dynamic snapshot of your immune metabolic balance. And in both scientific literature and also our internal data, we see that individuals with higher fiber intake, stronger omega-6 to 3 balance or better balance, and lower systemic stress, they generally show a lower kynurenine to tryptophan ratio, which is consistent with reduced immune strain and more balanced tryptophan metabolism. So each of the ratios I've talked about here, they represent a relative signal that shows whether your system is trending towards microbial cooperation or immune activation, which is based on patterns that are consistently seen in both research and our internal data. Now, when you reach the bottom of the report, you will see the quantitative metabolite values, which is the actual measured concentrations of IPA, kynurenine, and tryptophan. And these numbers are presented together with a target range. And I think it's worth understanding what this range really means. Because the first thing I want you to know is that there is a difference between what's biologically normal and what we consider optimized. So the normal values reflects what's commonly seen in the general population and in published research. So these are the ranges that you expect in an average population. But our internal reference group is based on people with high fiber intake and strong omega-3 protection, which consistently show tighter patterns that align with calmer immune profiles and stronger microbial activity. So these optimized target ranges are what you see on the report. So if your results fall outside of this range, it doesn't necessarily mean that something is wrong. It simply means that the biology hasn't yet shifted towards the optimal metabolic balance. So just to take an example, if a kynurenine level is 1.9 micromolar, this is entirely normal, but it is above our optimized target range, which suggests that the immune pathway is still moderately active. And this isn't necessarily a clinical concern. It just points to an opportunity for improvement through diet and through lifestyle. So these quantitative values, they can be viewed in two ways. First, as context, giving transparency and also allowing comparison with published studies. And second, as your personal baseline for future tracking. So even small shifts over time, such as stable tryptophan levels with rising IPA or a slight drop in kynurenine, will show measurable improvements in your gut immune cooperation. But in order to ensure an accurate comparison, the test should always be taken under similar conditions, fasted in the morning, and preferably not after heavy exercise or alcohol, because this will influence your kynurenine values. So in short, the values that you see at the bottom of the report, they are the real biochemical data, and the target ranges next to them, they represent what's actually achievable when the body and the microbiome is in balance. And again, the goal of this test is not to diagnose disease, but to guide, to help to translate the gut's chemistry into something that is clear and actionable. And this brings us to the next part, which is about how to turn these insights into action. Because the real value of this test isn't just knowing your numbers, it is understanding how you can influence them in the right direction. So, if your results show lower IPA, this suggests that your gut bacteria are producing less of indole metabolites like IPA. And this reflects that your microbial ecosystem, it may not be fully supported by your diet yet. IPA production depends on cross-feeding between specific gut microbes, which are fueled primarily by fermentable fibers and plant-based substrates. So when IPA is on the lower side, you should focus on feeding those microbes, increasing your intake of soluble and fermentable fibers from oats, barley, inulin-rich vegetables, legumes, and a diverse sources of resistant starch. And diversity matters because you should combine different fibers that creates overlapping microbial niches that will all boost indole production. And polyphenols, which you find in berries, olive oil, herbs, and tea, like I said earlier, these act like microbial selectors that will enrich the species that converts tryptophan into IPA. So pairing fiber with these plant-based compounds amplifies the effect. And of course, consistency matters. Your microbes adapt to what you eat most of the time and not what you eat occasionally. So you should aim for a steady daily fiber exposure rather than just occasional bursts. And of course, remember your omega-3 fatty acids, as they help to support balanced immune tone, balanced immune communication, which creates a gut environment that is favorable for indole producing species. Now, if your results show higher kynurenine, a higher kynurenine level usually reflects that the immune system has been a bit more engaged. It could be from inflammation, infection, stress, or lack of recovery. And it's part of the body's normal adaptive response. Kynurenine is produced when enzymes like IDO1 or TDO are activated, which is often by stress hormones or cytokines. But what matters is that how you help the system reset. So what helps here? First, you can support balanced immune activity through your diet by prioritizing omega-3 fatty acids from fish or algae or supplements. You should reduce processed oils that are rich in omega-6 and also include plenty of colorful plants that provides antioxidants and polyphenols. And you should also manage your physiological stress. Poor sleep and high psychological stress, as well as overtraining, it all pushes tryptophan towards kynurenine. So supporting rest and recovery and relaxation helps to shift your metabolism towards balance. And again, of course, fiber plays a role. Fermentable fibers and short-chain fatty acid production, it suppresses heightened immune signaling, which indirectly lowers IDO1 activity and therefore allows more tryptophan to reach the microbial indole route. So a higher kynurenine result isn't necessarily bad per se. It's a snapshot of a system under load. And this load can be reduced by nurturing the same inputs that will support IPA, which is fiber, omega-3s, antioxidants, and restorative habits. So what does this all mean in your daily life? It means that your microbiome and your metabolism isn't fixed. It responds to what you do, and every meal, every night of sleep, every walk outside helps influence this. So you should start simple by eating a variety of plant-based fibers every day, adding polyphenol-rich foods, balancing your fats, managing stress through recovery, mindfulness, or simply slowing down, and of course, sleeping well and moving regularly. And when you listen to all of this, they might sound familiar because these are all the fundamentals of a healthy lifestyle. So what this science shows is basically why those basics matter. They're not just generic wellness advice. They are the biological levers that will tune your gut and your metabolism and immune communication into balance. And of course, remember the goal isn't perfection. It's just to working towards progression. Even just small, consistent improvements in your diet and your lifestyle can help move these markers in measurable ways. So what this really represents is systems thinking in practice. We're not measuring a single molecule, we're measuring a pattern, which is the interplay between our microbiome, the immune system, and our metabolism. And each of the molecules are like data points, but when you view them together, they form a bigger picture, which is a system's fingerprint of how your body is functioning. And this is also what makes this so exciting. It's not about replacing medical diagnostics. It's about expanding our understanding of preventive health and seeing biology as a dialogue that we can influence through diet and lifestyle. So just to summarize, when tryptophan flows into microbial indoles like IPA, we see cooperation, we see balance and resilience. But when it flows too much into kynurenine, we see systemic strain. We see heightened immune activity and reduced adaptability. And because these molecules circulate in the blood, we can now read this balance directly, which turns this invisible chemistry of the gut into a measurable window on systemic health. So it's not just about the microbiome, it's about what it's saying and how we respond to what it's saying. And the gut health test gives us the first practical way to listen to this, to translate the gut's molecular language into something that is personal, actionable, and human. And this is also what makes the gut health test the next generation of gut testing, because it's translating cutting edge science into clear, personal, and actionable insights. And it bridges the gap between research and real life, giving individuals a way to understand how the choices they're making shape their biology. So in the bigger picture, it transforms complexity into clarity, moving from thousands of microbial names on a list to just a few measurable molecules that will reveal whether your system is trending towards protection or stress. And what makes this so powerful is that it represents a shift from observing disease to understanding health. So instead of waiting for something to go wrong, we can now measure how well our system is adapting, how balanced the dialogue between the gut, immune system, and metabolism really is. And this is the essence of preventive health. It's not about prediction of disease, but about detection of imbalance while it's still reversible. And the gut health test gives us a way to see whether the body's internal chemistry is trending towards protection or strain. And more importantly, how you can shift it back. Because when we are able to see change, we are also able to act on it. And that's how science becomes prevention and how prevention becomes empowerment. Thank you.